So Decipher is a free web-based platform. It helps clinical and research teams to assess pathogenicity and to share patient data, including both phenotype and genotype information. So Decipher is not about sharing the whole VCF from a patient, and not about sharing all of the variants. It's about finding those variants that are plausibly pathogenic, so those that are most likely to be causing the phenotype seen in your patient. So the aim of Decipher is to integrate the genotype and the phenotype data to determine the consequences of variants for health and disease. And this is terms for diagnosis and discovery. So just quickly, some Decipher highlights. So Decipher was established in 2004, so we've been running approximately 17 years. There's currently around 39,000 synonymised rare disease patient records that we share openly on the website. These contain more than 50,000 variants and more than 168,000 phenotype terms, mainly in HPO. We're used in more than 40 countries around the world, and there's more than 250 projects. So these are academic clinical genetic centres that deposit patient data to the platform. As well as open sharing, Decipher supports consortium sharing. So this is sharing between trusted partners, and we currently share more than 60,000 patient records in this way. So Decipher is a comprehensive interface in terms of genotype. So we support the deposition, interpretation and sharing of sequence variants, copy number variants, aneuploidy, UPD, insertions including mobile element insertions, short tandem repeats and inversions. We support both the nuclear and the mitochondrial genome, and everything is shown in a single interface. So this is just an example, and you can see that we've got a variety of variant types. They're all shown within the same page. Decipher is also a comprehensive interface in terms of phenotype. So most of the information is put in via HPO terms, but we also support some quantitative data. So we currently support milestone information and measurements, so growth measurements and visual measurements. We're very happy to work with other communities if they want to share this data. So we don't share the quantitative data on an individual patient basis. We aggregate it across genes, and I'll show you slightly later in the presentation how we aggregate this data. It's also possible within Decipher to add a disease. So if you have a diagnosis for your patient, you can say which diagnosis that is. Anyone can go to the Decipher website and they can search the openly available patient records. So this is the Decipher website here. And what you'll notice at the top, there's a search. There's many ways you can search Decipher, and this is just some examples. So it can either be by a phenotype, a genomic region, or a gene. You can also combine different terms. So in this example, I'm just going to add the gene EP300 into my search term. And what you get when you search for EP300, you get all the individual patient records who'll have a variant in EP300. You can see the genomic location, and you can see the phenotypes associated with that patient record. And decide which type of variants you would like to see, sequence variants or copy number variants. If you prefer, you can also see this information on a genome browser. So you can see here all of the individual patient records. To find information about the EP300 gene, if we go to this link at the top here, we go to what we call a gene page. So on these gene pages, we show all the gene disease associations. So these are from OMID, from Morbid, G2P, this is gene reviews, ClinGen, gene disease and dose sensitivity, as well as GenCC. And you can click on these information to find more information. So if we look at the ClinGen annotations, we can see the scores for haploinsufficiency. We can link out to ClinGen, or we can look at the PubMed IDs. As another example, if we click here for the GenCC data, we can see which disease were associated and how they've classified this gene disease interaction. If you scroll down on the gene pages, you can see the aggregated quantitative data that I mentioned previously. So in some cases, you can see a composite face. These are not automatically generated because we have to look at them individually to make sure that they don't look like any of the patients that go to make up that image, but they show the facial dysmorphology of patients you would expect to see with a pathogenic variant in that gene. We can also see lots of other quantitative data. So just to describe this first graph here, what you can see is plotted around the standard deviation. We can see the curve for the normal population, what's seen within Decipher, what's also seen for patients with a pathogenic or likely pathogenic variant in EP300. So all of these graphs are automatically generated in Decipher as soon as we have 5 patients with a pathogenic or likely pathogenic variant. If we scroll back up to the top of the gene page, you'll notice there's lots of other tabs with lots of other information. If we go to this tab called protein or genomic, what we see is a summary, a genotypic summary, and we can see the protein view. So just to describe this, in the center we have the PFAM domains. This is fully interactive. If we click here, we can see more information about this domain, and we can also link out to other resources. At the top, above the PFAM domains, we have all the Decipher variants plotted. We have the ClinVar variants all plotted below. The colouring is that anything in yellow is a missense variant, anything in red is a likely loss of function variant, and if they're filled in triangles, they're annotated as pathogenic and likely pathogenic. At the top of the screen, we can see exon information. We can also see a predicted NMD escape track, and we can see various information from Nomad. You can click, if you so wish, on any of the Decipher variants to find further information about the patient, and you can actually go to the patient record if it's of interest. Also available, you'll see there are 3D structures. And if we click on these 3D structures, what you can see is the 3D structure, and you can see all the Decipher variants plotted within this 3D structure. You can click on these to see which patient record they refer to. Another tab that we provide is a phenotype tab. So what we're seeing here, this is again for open access patients in EP300. We can see all the phenotype terms associated with the patient records. So what you can see is the code, the phenotype, the affected patients and the percentage of those in EP300. And you can compare this with what's seen across the whole of Decipher. And what we can do is sort this with the p-score. So what we've done here is we found the most discriminatory phenotypes are at the top. And in this way, we can use this aggregated data to find this type of information. Also available is a matching patients interface. So what we're seeing in this interface, so in this case, all the sequence variants associated with EP300. We have some summary information such as consequence, inheritance and pathogenicity. We also have this list which shows us all the phenotypes that are present in multiple patients that have a variant in EP300. We have all the individual patient records at the bottom here. You'll notice there's a series of filters here, and what we can do, we can filter and drill down into the data that we really want to see. In this case, I'm just going to show the variants that are pathogenic. And you'll notice when I select these filters, all the information in the summary, this list, and also the individual patient records are all updated. If you find in here a patient record that you're particularly interested in, you can use this contact button here. You can actually send the depositing clinician a message about a patient match or a possible collaboration. So obviously it is possible for you to deposit data to Decipher, and when you deposit data you get additional interpretation interfaces and you get access to more features. So I'm just going to show you some of these features. So obviously what I've shown before is we support the deposition of all types of genetic variation. How you deposit this via the website is there's a form for each type of genetic variation, and you just complete the form. What I should say, if you have lots of data to upload, there is a bulk upload option. So you can upload the data as a CSV file or an Excel file. We also have a deposition API. So Decipher visualizes all the genomic data in build 38, but it's possible to deposit the data in build 37, and Decipher will lift that data over to 38. The other thing I'll mention is there's actually 3 sharing levels in Decipher. So you can keep the data private, so it's only available to those users of the depositing centre. You can consortium share it, so that's sharing with your trusted partners, or with explicit patient consent, you can share the data openly on the website. And the way that this is controlled is each variant, you can select the sharing level. So in theory, you could have for one patient a variant that's private, consortium shared, and an open variant. So the depositing centre is very much in control of what data they're sharing and how far and wide they're sharing the information. So in terms of depositing the phenotype information in Decipher, there's an interface to allow you to find the relevant HPO terms. So all you do here is add your term. And when you add your term, you find that all the matching terms are shown. If you want to look further down the HPO tree, you can just click here and actually look further down the HPO tree. I'm going to go back to the search results, and all you do to add a phenotype is press the present button here, then moved over here onto the patient phenotypes. So you can very quickly phenotype your patient. If relevant, you can also add phenotypes for the mother and father and add any other relevant family members as necessary. It's also possible to add some measurements, and if we go to the measurements, you can just click on here, and the types of measurements available are listed. You can select the one that we would like to add, and there's a simple form to fill in. And once you've added the information, you get a graph. It shows you where your patient lies compared to the median and the minus and +2 standard deviations. In this case, we can see this patient's about -2 standard deviations from the norm. The milestone information is added in a very similar way. We just click on the edit button, we add the milestone information and we save, and then we've deposited this information. When you deposit variant data to Decipher, in this case a sequence variant, you get some annotations. We know that it's a missense variant. We can see that it's not in Nomad. And what you'll find is you get a variety of interpretation interfaces that directly relate to your variant. So the first one that I'll show you is the browser here. So the browser is fully interactive. We have filters on the browser. So in this case, if we're looking in the genes track, we can choose if we're seeing protein coding genes, non-coding genes, or all genes. We can also decide how we'd like the genes coloured. So in this case, we're colouring by PLI score. Everything in the browser is interactive. So if you click on, for example, missense constraint, there's a pop-up that shows you actually the missense constraint information. If you're looking at a different type of variant, such as a CNV, you again get a genome browser. It has slightly different tracks because obviously you're more interested in larger, in most cases, larger variants. So in this case, we have a Decipher population copy number track, a DGV gold and Nomad structural variant, a ClinGen dose sensitivity region, and also a ClinVar structural variant map. We can also obviously see all of the Decipher copy number variants. For CMVs, you also get a tab called Genes. So this lists all of the genes that your CMV overlaps. You can use this in a variety of ways. You may decide that you want to sort it, i.e. In this case by the PLI score. So we're bringing the gene that's got the highest PLI score to the top. Or you may wish to use the filters at the top here. And in this example, we're just filtering for anything that has a ClinGen annotation. And in this way, you can easily determine how many protein coding genes you have in your CMV that overlap, and also if there's any relevant genes. So as previously, you get a protein view, but what you'll notice in this case, because we're looking at a patient record, we can actually see via this line exactly where our patient's variant is lying on the protein. Again, if you click on the 3D structure, you can see the 3D structure and you can see your own patient's variant. So coming very soon for small CMVs, you'll be able to also see how the protein's affected. In this case, this variant is a deletion, and you can see which area of the protein is deleted in your patient. There's an annotations tab. So this is an example of a sequence variant. So for sequence variants, we run the ensemble variant predictor across all of the different transcripts. And we also provide various annotations. So we provide SIFT, Polyphen, CAD, Rebel, and Splice AI annotations. What you'll notice here is that we also tag main select transcripts. So main is a project between ENVIL-EBI and NCBI, And all of these transcripts match the ensemble transcript as well as RefSeqs. They match the genome, build 38 genome, and they're a primary reporting transcript. There's a tab that shows you the allele frequency in Nomad, and alongside this we have a tolerated population variation calculator. So this is work by Nikki Whiffin, and this calculator can help you determine if a variant observed in a reference sample is too common to give a Mendelian disorder of interest. Again, you can look at the matching patients interfaces. Because we're actually in a patient record, you're getting some extra information. What you can see is your own patients' phenotypes that are present in other matching patients. And you can also see your patients' phenotypes that are absent in matching patients. You can also see the copy number variants, matching patient interface. It's very similar to the sequence variant interface. It just has some slightly different filters and different summary information. We also have a tab that's called functionally identical variants. So if your patient's variant has been seen in other patients, you will see this tab. It helps to bring the information together for that specific variant. It helps you to look at the phenotypes and also to see how other centres are scoring that particular variant. If we look on the Genes tab and we look at the quantitative data, because we have right access on this patient record, we can see exactly where our patient lies in the distribution. So, for example, here we can see the head circumference and also first words. So this helps to decide if you are looking at a relevant variant. Obviously, in this case, our patient lies within an expected distribution for a pathogenic CDK variant. But for instance, if your patient actually had macrocephaly and all the other patients had microcephaly, it would help to suggest that maybe this particular variant isn't relevant. So Decipher is part of the Matchmaker Exchange, and within Decipher, you can click on this tab and you can query the Matchmaker Exchange. So the Matchmaker Exchange is a global alliance project, and it helps find other patient matches. It's a federated query system. So if you're within Decipher, and what you can do is send a query to other related nodes, and then if they have patient matches, they'll bring that information back into the Decipher interface. And what you'll see is a big table that shows you all the potential patient matches. So once you've used the interpretation interfaces to look at your variant, there is a pathogenicity evidence tab. So this is the case for copy number variants and sequence variants. In this case, the sequence variant module. So we have available evidence types on the left, and we have selected evidence on the right, and evidence to consider here. So you can classify your variant according to ACMG standards. So all you do is you scroll through the different available evidence types and you click on the criteria that are relevant, and it's moved over to selected evidence. What you'll find in some cases, there's a button, a link here that says further information. And the information pop-ups show you when ClinGen has recommendations for how to use a particular criteria. In this case, it's PVS1. We actually have an interactive PVS1 tree that you can use to decide if you should keep the same evidence level or if you should modify the strength. So in Decipher, to modify the strength, all you do is click on the relevance criteria and select the strength from the drop-down. In this case, I'm going to modify it to strong. Once you've added all your relevant criteria, we tell you how the variant will be classified according to the 2015 ACMG standards. In this case, it's pathogenic. We also apply the ClinGen Bayesian classification framework, and in this case, it's pathogenic. We show the posterior probability. You can then select the pathogenicity and contribution. There's a notes field if you would like to add any information for other users in your centre to know why you've added the criteria that you've had. And once you've saved that information, there's a summary screen that shows why the criteria that you've added and how you've classified your variant. In some cases, ClinGen have expert panel specifications, and if they're available for a gene that you're interpreting, we do provide a link and you can go to these specifications when you're adding pathogenicity evidence. We've recently released a similar interface for CMVs. It's set up in a very similar way. So we have available evidence types on the left, selected evidence on the right, as well as evidence to consider. Again, there's some further information pop-ups, and when you click on here, you get further information about how to use the criteria from the original paper. You can modify the strength just by clicking on here. You can either up or downgrade the strength. So once you've added all the relevant criteria, again, we tell you how it would be classified according to these technical standards, and we provide the score. You can again select the pathogenicity and contribution and add any pathogenicity notes as required. And obviously, you get a summary of this information once you've saved it. So Decipher is completely free to join, and to be able to join the Decipher community, you need to be an academic centre of clinical genetics. We support both clinical and research projects, and it's completely free to join. To join Decipher, all you do is click join at the top of the web page, and there's two different ways to join. You can either join an established project with a very short application form that you need, or you can apply to set up a new project if your institution doesn't yet have a Decipher project. So what I'd like to do is sort of end where I started. So the aim of Decipher is to integrate the genotype and the phenotype data to determine the consequence of variants for health and disease, both for diagnosis and discovery. And what I'd just like to point out is Decipher has very granular phenotypes. So this not only helps to inform the diagnosis and helps you to decide if your patient really is a very good patient match with another patient in Decipher, but it allows you to find new phenotypes associated with a disease, and it also allows you to find new variant disease pairing. So as you will be aware, in some cases, depending on where the variant is within a gene, depends on which syndrome your patient has. So there may be several syndromes associated with a particular gene. Decipher really does fuel rare disease research. Since its inception, there's been more than 2,600 publications citing Decipher data. So I'd just like to give some acknowledgements. We have patients and families for permission to include their data in Decipher, members of the Decipher community, as well as the resources which Decipher uses. This is the team here. Matt Hurl's our academic lead, Helen Firth our clinical lead, and our contact details are here.